I got loyalty inside my DNA. How's it going there, barcoders? So today we're going to work on activity 3 in the PCR lab, figuring out how to align primers in the genome of our target organisms. All right, so we're working on activity 3 here now, and the first thing that you're going to want to do is just download this APE software. There should be a link to that in Module 6. So let's see here. Here's Module 6 on Canvas. Here's download latest version of the APE software to your laptop. So go ahead and just download it. In this case, I'm doing the Mac version. Open that up. Drag and drop it here. Okay, so now I've got the APE software and I can go and find it. Let's see here. Under applications, APE. So I'm going to go ahead and open that up. And you may get a message that says this cannot be opened because the developer cannot be verified. If that happens, go ahead and just click this little question mark here. And then hit the Open General pane for me. Go ahead and click right here. And then you'll get something with security and privacy. And it basically says, hey, can I trust this software? Let's try this again. So open up APE, do that. Open the general pane for me. Now it says APE was blocked from use because it is not from identified developer. Go ahead and hit the Open Anyway button. And then I'll close out of this. Hit the Cancel button. And let's try again to open up the APE software. Applications, APE. Now we can hit Open, and now we can open up that software. Okay, so the first thing that we're going to want to do in this activity is to identify the genus and species of the organism that you're trying to amplify. So in this case, I know that I'm looking to see whether vegetarian hamburgers are contaminated with beef. And so I have to figure out what is the actual genus and species name of beef. So I'm just going to put in cow, genus, and species. And I know that it's Bos taurus now. BOS is the genus, taurus is the species name. So I'm going to go ahead and write that down in my notes. And then the next thing I need to do is figure out what is the actual DNA sequence of the region that I'm trying to amplify within the Bos taurus genome. So the way you go about doing that is going to this thing called the NCBI GenBank database. So there's a link here in the lab manual that you can click on that will take you to this site. And essentially what you're trying to do is look up what is the actual genetic sequence of Bos taurus in the mitochondrial region that we're trying to amplify. So how do I know it's the mitochondrial region? Well, it really depends on the primer set that we're using. So I know that Bos taurus is a vertebrate and it's an animal, and I know that for vertebrate animals, the barcoding region that we use is the CO1 or cytochrome oxidase 1 region, also known as the COX1 region. And so we can use this table here in the lab manual printout, depending on the different taxonomic group that you're trying to search the gene for. And basically, we're going to use this information because we have an animal. And so we're looking for this information. What is the COX1, the cytochrome C oxidase 1 mitochondrial complete genome? Searching any of these terms will help us find the specific sequence of DNA that we're looking for our target organism. And you want to also make sure you put in the full scientific name and the word complete. So let's go ahead and do that on this. So we're looking at, we've got nucleotide selected here. I'm going to put in the genus and species, Bos taurus, of the organism I'm looking for. I'm going to put in COX1, which is the region of the genome that I'm looking for. And then I'm going to put complete to say that I only want to find COX1 regions that are completely sequenced. I don't want partial sequences. I want the complete sequence. And then I'm going to hit the search button and see what pops up. Okay, so here we can see we've got Bos taurus. So this is all these different hits represent different entries into the NCBI GenBank database. We've got Bos taurus, we've got the mitochondrial, and we've got the complete genome of the entire mitochondria. So this is the whole mitochondrial genome as opposed to just the COX1 region that we're looking for. And that's totally fine. That'll work. It's not too much to copy. So I'm going to go ahead and hit this FASTA button. And here this will show us the actual DNA sequence of the complete Bos taurus mitochondrial genome. Do note that this information here is ancillary information. So this is the GenBank unique accession number. So this is one individual cow whose mitochondrial genome was sequenced. And this is basically a unique identifier for that cow. This is the genus and species. This is the fact that it's a mitochondrial genome, and this tells us it's the complete genome of the mitochondria from that specific species. So I'm just going to highlight all of this sequence. I'm going to copy it, and then I'm going to paste it into the Ape software. So I'm going to go ahead and hit paste. Now I've got the whole mitochondrial DNA sequence here in this Ape window. So one thing that we can see here is this will tell us the total number of base pairs in that sequence. So in this case, we have 16,340 individual base pairs, and that should match the information that we have here. So if I go back, you can see that it's 16,340 base pairs. So that's good, that information matches. So now what we're trying to do is figure out, okay, within this mitochondrial genome, are our primers going to bind to a specific region within this mitochondrial genome? Are the primers that we're using going to work? And are they only going to bind to one region? Are they not going to amplify multiple regions within the genome? So basically what we need to do is, number one, figure out what the primer sequences are, and number two, figure out, do those primer sequences align or complementary base pair with any particular parts within the mitochondrial genome of a cow? Okay, so in the lab manual, you can see that there's a list of all the different primers that are available to us. And in this case, we need to figure out which primers did we use in our PCR. So hopefully you guys use the MTCO1 primers, specifically the non-fish vertebrate primers. So I'm going to go ahead and highlight or copy this sequence of the forward primer. So I'm going to highlight, remember, not the 5' and 3', just get the actual sequences. And I'm going to hit copy. And then what I'm going to do is use this Ape software to search within this entire mitochondrial genome sequence to see whether or not there's a spot where the primers will bind. So I'll hit edit. I'll hit bind. And then I'm going to paste in my sequence. And it's very important that before you do this, you make sure that your cursor, so let me close this, you make sure that your cursor is at the beginning of the sequence right here. So go ahead and click it and make sure you can see the cursor flashing there. So once again, I'm going to go to edit, find. I've got my DNA sequence in there. And then I'm going to hit this button, find next. And what you can see is nothing's happening. So one thing that this allows you to do, it says allow mismatches, and it's set at 0. So right now I'm saying, is there anywhere in here where this primer set matches absolutely completely without any mismatches? And saying no, there's not. And really you can go all the way up to probably 4 or 5 mismatches, and the primers will still bind pretty well in your PCR. So let's look at whether or not it works for 1 mismatch. Nope. What about 2 mismatches? Look at that. So now we've got this highlighted region, and basically our primer will anneal to this region but there will be 2 mismatches. So what I want to do is go ahead and actually mark that region. So this is the spot where our forward primer binds. So I'm going to hit close here. I'm going to hit feature, going to hit new feature, and then I'm just going to hit the OK button. So now that region where the forward primer binds is highlighted. So the next thing I want to do is look at whether or not there's a spot in the genome where the reverse primer will bond. So I'm going to copy the reverse primer sequence. I'm going to once again go to find. I'm going to paste in the reverse primer sequence. You want to make sure that this little box here is checked that says also find reverse complement of the string. I'm going to hit find next. And here we've got it. Let's see if it'll go all the way to 0 mismatches. Find next. No. 1, 2. Okay. So once again, we've got a match with 2 mismatches here. I'm going to close this. I'm going to hit feature, new feature, and hit OK to add that feature. And so now we've got where the forward primer binds and the reverse primer binds. And so what we can do here, or what we've learned from this, is 3 things. Number 1, our forward primer binds at a decent amount, an allowable amount, so it'll bind to the sequence with only 2 mismatches, which is sufficient to say that we can feel confident that our PCR will work. In addition, the reverse primer also binds with only 2 mismatches, which is a sufficient amount to say that this primer is probably going to work. And we also know all the DNA sequence in between, and we can actually use this to figure out what's the length of our amplicon that we're going to get when we run this through a gel. So our amplicon is going to be from the start of the forward primer to the end of the reverse primer and all the DNA sequence in between. And if you look up here, you can see the length is 709 base pairs. And so we know that our primers will anneal properly, and we also know the length of our amplicon if we're amplifying Bose-Torus mitochondrial DNA or cow mitochondrial DNA. So if there is beef present in the vegetarian patty that I'm looking at, then there should be Bose-Torus or beef mitochondrial DNA in that beef patty. The primers that I used should anneal to that DNA, and we should get an amplicon with a length of 709 base pairs. So what this means is if the PCR fails, it's not because our primers aren't well designed. If the PCR fails, it's because that DNA probably isn't present there, or there's something going on with the master mix, or there's not enough template, or there's impurities or a bad concentration of DNA. So what you can do with this once you're all done is you can take a screenshot of it. And you can paste this into your lab notebook. And make sure that that screenshot includes the region that you're amplifying so you can see the length very clearly in that screenshot. And each group member can do one of these. So each group member has a sample. Make sure you include the genus and species of the organism you think you'll find in each sample and your predictions for whether your primers are well designed or not, and your prediction for the base pairs or the pattern of bands you should see in your gel.